we do agree that sponsors can supplement with retrospective samples, but usually only after conducting a prospective study, which yielded too few samples to demonstrate adequate performance. In terms of U.S. versus outside the U.S., we recommend that sponsors conduct their prospective clinical studies primarily in the U.S. to support a 510K or de novo, but data from outside the U.S. Prospective clinical studies can also be submitted to supplement the U.S. data, especially in cases where there's difficulty getting enough specimens in the U.S. Due to low prevalence with a heavily vaccinated population. Our next question also has to do with imaging studies due to the current COVID positive cases decreasing here in the U.S. And mentioning a comment that Dr. Stenzel made on a previous town hall about the acceptability of enriching studies in order to meet the study requirements. The question is asking if we can clarify what would be appropriate for study enrichment. So there are multiple options for clinical study enrichment, and they really should be appropriately tailored for your specific test and the claims that you are validating. So we do recommend that you submit a pre-EUA so that we can review your proposal and consider the alternate study designs that would streamline enrollment of positive subjects when prevalence drops low. That would be best appropriate or most appropriate for your situation.
Yeah, and thanks, Debbie. And I would add that the original reason to offer enrichment was for stimulating additional development of screening assays. So for screening asymptomatic patients, and when sometimes in populations that's very low, or you have to screen a lot of folks, we have now considered doing to allow enrichment for original submissions where obtaining a positive may be more challenging, symptomatic positives even, and where maybe bank samples aren't appropriate or as such. But we would still like to see a prospective attempt to try to get true positives, and then looking at supplementing them with a method of enrichment. Bank samples is one. The other is to use some sort of selection where a patient may be, and this might be more so for an asymptomatic carrier than a symptomatic person, but it could also be done with a symptomatic person, and that is where they are identified as positive with some orthogonal method and some sort of routine study or protocol for screening. We're open to study designs that try to mitigate the bias in that situation. If the patient themselves knows the result of the test and it's not going to involve self-collection for the candidate test, then that may be acceptable. If the patient, though, is going to be doing self-testing with candidate device, knowing the previous result, will bias the result. But for the situation where they're not going to be self-collecting for the candidate device, the person doing the collection, should be blinded to the previous results. Patients should be asked not to identify whether they're positive or negative, and of course to keep the administration of the study honest and unbiased. Suitable number of negative patients should be offered in the same way, so that the person doing the testing doesn't assume that it should be positive. This way, bias risk can be mitigated to a great extent. So there are multiple paths, and we're open to multiple paths. We do highly recommend that you run those strategies by the FDA prior to initiating your study, in order to avoid any sort of duplication of studies, because the study plan and design was not suitable. Back over to you, Toby.
Thanks, Tim. That's really helpful to add that additional information. Our next question is regarding a professional use rapid antigen test. This sponsor is conducting a clinical study and has so far collected some positive samples, but none of them have CT values over 30. They're looking for FDA's feedback on what to do if they're still not able to get any or enough low positives, since FDA generally does want to see 10 to 20% of positive samples being low positives with a CT over 30 for antigen tests. So we do generally still need to see at least 10 to 20% of low positives as observed in other sequentially collected prospective studies. But we are happy to discuss enrichment strategies, as we just talked about, in a pre-EUA. So we would request that you submit a pre-EUA with your proposal, and we can give comprehensive feedback that way. The next... go ahead.
If I could add just a little bit to that. So
yeah.
One thing with an antigen test device, if you're skewing patients towards the early days of symptoms, that may have an effect on comparator CT values. They would expect it to be higher once a PCR test is positive. So we look at the scatter of patients across the window of claimed days of sensitivity, and it may be that collecting more samples further out from symptoms in the study is a way to enrich. But if you're in this situation, we do recommend you come in with the pre-EUA and discuss your challenge and potential solutions with the FDA team. Thanks. Back over to you, Toby.
Thanks, Tim. So the next half of this question is about, so for the same rapid antigen test, asking about usability in clinical studies for non-prescription over-the-counter use. The question notes that a couple products were recently granted EUA for over-the-counter serial testing. So they're asking questions about whether the usability study and the clinical study need to consider serial testing in the study protocol design. So I want to make sure that everyone, including the sponsor, is aware of the supplemental template for adding serial testing. That was put out in March, I believe, and it does lay out exactly what situations we think we can add serial testing, including over-the-counter serial screening without having prospectively collected data on asymptomatic individuals or for serial testing specifically. So for the initial authorization, we would not expect to see serial testing specifically evaluated in your usability or clinical study. We would expect you to follow that, the antigen template and then that supplemental template, which indicates that you would need to demonstrate a PPA of 80% with a lower bound of 70% for individual single-use testing on symptomatic individuals. And based on that performance, we would be able to give you the indication of serial testing for asymptomatic individuals with a post-authorization requirement to validate the asymptomatic serial testing claim. and that would be through a protocol that we would review interactively with you before you implemented that as your post-authorization requirement. All right. You did have another question. Oh, go ahead.
Yeah, what is the next question? You want to just read the next question because I may be answering one of these that are upcoming. Is it the EUA letters of authorization question that's next?
Yes, there's a question about EUA letters of authorization containing several statements that are required to be included on all descriptive printed matter, advertising and promotional materials. And the question is asking for online advertisements. Is it acceptable to omit the statements due to space limitations if a link to a web page with the full list of statements is included in the online advertisement?
Yeah, Toby, I didn't see a prepared answer, so I thought I could jump in unless you have this one.
I think we were planning to get a written response for that one to make sure that we get all of the legal nuances on that one, actually.
Yeah. Okay. Well, my suggestion was going to be that that is in the letters of authorization. We generally expect that to be followed, but if there is some sort of situation where it's hard, certainly we're open to different ways of handling it. So I would approach the FDA on that. But typically we want all the important information on our material. Thanks, Toby.
Yeah, great. Thank you. All right, let's see. The next question is about a 510K for molecular SARS-CoV-2 RT-PCR with claims for asymptomatic population. And asking FDA's thoughts on comparator method for that study, specifically whether the comparator assay needs to have asymptomatic claims and whether the comparator assay needs to be cleared, and as well as whether the BioFire RP 2.1 assay is an acceptable comparator. So we have not, we don't currently have any tests that are cleared with an asymptomatic claim for SARS-CoV-2. So at this time, we would recommend that you use a comparator method consisting of 2 EUA-authorized, highly sensitive RT-PCR assays that do have that asymptomatic claim with a third test available as a tiebreaker. And as we've discussed previously, we do recommend that you submit a pre-submission to discuss your proposed comparator method prior to moving forward with that towards the 510K. And our next question is about clinical evaluation of a point of care or over-the-counter antigen test for an EUA, asking about, due to the declining number of positives in the U.S., whether some or all of the study can be completed outside of the U.S. So we do, it is acceptable for some of the data to come from prospective clinical studies outside the U.S. to supplement a U.S.-based prospective study if that U.S. data is not sufficient due to low prevalence in the U.S., since we do have a heavily vaccinated population now. However, we would need to understand if the testing is being conducted under the exact same protocols and whether the population tested appropriately represents the U.S. population. So we would recommend in that situation to submit a pre-EUA to discuss the proposed clinical evaluation to make sure that it's appropriately designed and carried out, and we can give comprehensive feedback to you that way. Our next question is for, there's 2 questions, one about an antibody test asking whether a prospective study is required for an antibody de novo or 510K submission, or whether a retrospective study can include some samples from outside the U.S. So we do recommend that sponsors conduct prospective clinical studies primarily in the U.S. to support a 510K or de novo. Again, similar to the previous question, data from outside the U.S. prospective clinical studies can be submitted to supplement the U.S. Prospective data. And if site collection sites outside of the U.S. are chosen, you should document the relevance of the studies to the U.S. clinical practice and demographics. And then the second part of that question is about an antigen test, de novo or 510K submission, when only claiming use with nasal swabs, whether the comparator method can be a high sensitivity RT-PCR with nasal swabs, or does it need to be an NP swab for the comparator? So we have been accepting nasal and mid-turbinate swabs for the comparator method for EUA submissions, and I expect we would do the same for a de novo or 510K, but we do recommend that you submit a pre-submission again to discuss that study design so that we can provide comprehensive feedback.
And one sample type, Toby, that doesn't follow that recommendation is saliva, where we still recommend an NP swab, but for saliva, we'll also entertain a mid-terminate swab.
Great, thanks. All right. Speaking of saliva, our next question is about saliva, noting that on previous town halls we've mentioned that saliva is a challenging sample type for antigen tests, and asking whether we could explain more details on what issues were uncovered. I do want to note that we have mentioned that saliva is a challenging sample type both for molecular and antigen tests, not just antigen. And then the question also notes that FDA mentioned that freeze-thaw processes increase sensitivity in saliva antigen tests, and asking if this is the case for antigen tests detecting the N protein or the S protein or both. So we have seen that saliva is a challenging specimen type. Detection from saliva is typically less sensitive than upper respiratory specimens. The freeze-thaw issues can affect the samples in different ways, and in some cases we've seen where it increases, in other cases decreases. And so we have seen some variability in the data that we've seen from test developers with saliva in terms of the ability to appropriately validate it and to get appropriate performance with saliva. Tim, do you want to add that more?
Yeah, I would add that the freeze-thaw cycle, we recommend it be investigated if you're going to use frozen samples for any EUA authorization, to show that there is no bias in using frozen samples. Okay, thanks. Back over to you.
Great. And then the last prepared question that we have is about submitting an EUA for a point-of-care molecular diagnostic test system with an optional app to communicate the results of the test to the appropriate authorities if the user desires. Asking whether it is a requirement to include validation data for the optional app as part of the initial EUA submission, or if use of the app could be filed separately, or whether or not it is even necessary to provide validation data for the optional app. So for that, it would depend a little bit on the role of the app and how it interacts with the test itself. So that's something that we would likely want to have a more in-depth conversation with you about, so that we can better understand the interaction between the test and the app before we provide that feedback. So
Toby, there was
with a pre-EUA. Oh, sorry, go ahead, Tim.
I just want to wait until you're done. You done?
Yep, go ahead.
Okay, I just wanted to say that due to my fault, we passed over one of the questions earlier. I want to go back to it and answer it. And the question has to do with when does the FDA anticipate an FDA recommended reference material is available for antigen tests? And at this time, we don't have such a recommendation or a panel. There are clear challenges in doing this for antigen tests, and currently we do not have anything stood up. It would be great. So we're obviously thinking about it, but nothing right now. Thanks. And Toby, I think that might be the end. And can we go to the open line for questions?
Yes.
Okay. Back to you, operator.
Thank you. And at this time, if you would like to ask a question, please press star 1. Please unmute your phone and record your first and last name clearly when prompted. Your name is required to introduce your question. To withdraw your question, you may press star 2. Once again, at this time, if you would like to ask a question, please press star 1. And our first question is from Alex. Your line is open.
Hi, this is Alex with UserWise Consulting. We specialize in human factors and usability testing. My question is about inclusivity testing. Is there any expectation that manufacturers of OTC antigen tests include new recent strains of the virus in inclusivity testing, or can they rely on very common strains sourced from Europe, for example? And can you generally explain your latest expectations on the quantity of strains, and which strains should be used for inclusivity testing?
Yeah, Toby, you can correct me if I'm incorrect on our guidance related to variants and mutations. Our intention there was for developers to take a look at the variants in the U.S. population when they do their validation tests, and they determine whether or not there might be an issue with certain variants. If, bioinformatics analysis, you see where the mutations are, how your, what antigen your antibodies were raised against, so at least the area of the virus that you should look for potential, issues with certain mutations and variants. And apparently, with one of the antigen tests, there has been, fortunately, a low abundance mutation in the United States that we don't expect to increase, but has been in studies shown to not detect a particular mutation, virus with a particular mutation. So this has been proven to be real already for molecular. It's now proven to be real for antigen. Serology may be a little bit more insensitive to mutations and variants due to the polyclonal nature of immune responsive. And that they may help, but there should certainly be clear examples that come up for serology as well. So we are currently surveying for tests on the market, molecular, antigen, and serology. Information that we received from the developers having to do with how their tests were designed. It allows us to do a first pass, at least for antigen and serology tests for potential issues with inclusivity, false negatives. And we are currently reaching out when we have potential concerns and engaging those developers. And it is important for those developers to get their hands on some sort of a protein molecule with those variants. We're open to different wet methods to do that. I would suggest that if you do determine that there could be false negatives with overall at or above the 5% mark in the U.S. population, or a single variant or mutation that's at or above 5%, that you engage with the FDA on your strategy to assess that pre-authorization, so that we have good assurance that it's not going to affect performance of the assay. We also have in that guidance asked that you come in with your EUA, that you come in with a plan for how you're going to, on an ongoing basis, assess the risks of false negatives due to variants and mutations. And we'll look to that and give feedback on that, because that will be a post-market commitment that we will require for developers to monitor that situation. So, be anything to add?
That's just in addition to what you were covering. I would also note that the EUA templates do include recommendations for inclusivity testing. So I would also suggest that you take a look at the template for the specific type of test that you're looking at.
Thank you.
Thank you. Our next question is from Shannon Clark. Your line is open.
Hello, this is Shannon Clark with UserWise Consulting. Thanks for that really helpful response just now. Another question. My understanding is that in order to obtain over-the-counter designation for an antigen test kit with a mid-turbinate swab, the FDA requires inclusion of a stopper for child safety to prevent overinsertion. First of all, is this true? And then is the purpose of this stopper to prevent growth overinsertion, such that the stopper could be located 4 centimeters from the tip? Or does it need to prevent insertion past sort of the zone for 2-year-olds, which is about 1.5 centimeters or a half inch? What is the purpose of that stopper, and is it required?
It is safety related, and we'll take this on a case-by-case basis. For example, we've had some developers come in and say, that they're going to require a mid-turbinate swab, and it's sort of in the gray zone just beyond an anterior nares, sort of in the 1 inch or 1 centimeter, I forget. Versus a true, right up there mid-turbinate swab. But it is a safety consideration, so we will take this on a case-by-case basis per the device and per the device instructions. So if there's any questions or potential concern about that, engaging with the FDA early is good prior to doing your user usability studies, and perhaps also your clinical studies to make sure that it's done in a safe manner.
Okay, thanks so much.
Mm-hmm.
Thank you. Our next question is from Jackie Chang. Your line is open.
Hi. My question is about the expectation for a fully quantitative serology test, and also for a fully quantitative neutralizing test. Right now, the 2 templates are for semi-quantitative tests. And I'm just wondering if the validation requirement are just doing everything that is listed for the semi-quantitative test, and then also establishing traceability of the results to the WHO standard.
Yeah, establishing traceability is important to the international standard, reporting out in international units. But also, linearity and other issues. So I believe we've authorized 1 truly quantitative test, if I'm correct. So there is some information in that online information about that test, so I checked that out. We are working in this area. It is an area of importance and focus for the FDA. In the serology team, it's neutralizing antibodies and fully quantitative antibody assays are our current top priority, high priority for serology tests. So if you have, you can submit a pre-EUA and put in there what you think based on the prior authorization and how you want to plan it, and then we can provide some feedback on your validation study plans and what our recommendations are more specifically for your device.
Thank you. And just to clarify, we've authorized semi-quant. We have not authorized a fully quantitative test yet. but we do we are happy to help provide feedback through the mailbox or through a pre-EUA.
Yeah, so I submitted
so
a question to the mailbox.
That's the way to go.
I submitted a question to the mailbox, and I haven't received any answer yet, but I really appreciate this answer. Maybe...
So, yeah, send it back through and copy Toby. Ask them to copy Toby and me when they reach out to the team. Because the team has put together their thoughts on fully quantitative, and obviously we've been thinking about it and engaging with sponsors already for me to think we had fully quantitative already.
Okay, thank you.
Thank you. And as a reminder, if you would like to ask a question at this time, please press star 1. Our next question is from Kodumodi Venkat. Your line is open.
Good afternoon. Thank you for taking the question. This question is related to the serology test. I just wanted to find out, in last year, there was an umbrella EUA for serology tests, and then the independent validation was done at NCI. So my question is it the NCI has done all the validation, or still that independent validation is in effect? If you can just give me an update, I'll appreciate. Thank you.
Yeah, the NCI is still open for business, as I say, for those assays that the FDA prioritizes for the NCI. And we've asked the NCI to stay through a period of time where assays are converted to full authorization, or we receive novel applications through either de novo or a 510K pathway following the first granting of a de novo application for serology.
And just to clarify about your question about the umbrella EUA, there was an umbrella EUA issued last spring, and last summer it was revoked. There were no tests that were added under it, so it did not impact any specific tests, but the umbrella EUA itself was revoked, and we have been using the individual EUA pathway instead for serology tests that have, including the ones that have been evaluated by NCI.
Yeah, thanks. My question was something similar to what Tim answered. Is still the NCI is doing the independent validation. The question is, has the backlog of all the tests that were submitted they have done, and then the reports have been given, or still there are things that are being done at the independent lab?
That's a great question. I don't have an answer for that today. I know that that we constantly look at priorities, and if there is a priority, we do let the NCI know. So, but I don't. I haven't tracked lately with that backlog. I can't respond directly to that at this time.
Thanks.
Thank you. Our next question is from Shannon Clark. Your line is open.
Oh, hello, Shannon Clark. So a question about vaccination. So obviously you can't run a clinical study for a serological test kit with vaccinated individuals, as vaccinations may influence the presence of antibodies. However, can you include vaccinated individuals with an antigen clinical evaluation study?
Toby, I believe for antigen, yes. But we would want the data separated out with those who have been vaccinated versus those who haven't. The performance could differ, and in particular, if someone is vaccinated, they might have lower levels of virus in their nasal passages, and it could potentially impact the performance of the antigen test. So we don't. I'm not familiar with any data that we in vaccinated information. Presumably some researchers are doing that out there. Probably should ask our Antigen team if they've seen that yet. we're sometimes so busy we keep our nose to the grindstone with the applications. But it's a good question. And it's just a word of caution that vaccinated people may not behave the same way on a symptomatic follow-up infection as a primary natural infection.
But this doesn't necessarily double our sample size. We just can collect the data and then analyze the data separately if needed.
Yeah, you may find that it really drops your performance there, and we'd like to see the data in the vaccinated population. If it doesn't really drop the performance, we just want to see the data also separately. We may list it separately, together and separate in the IRB.
Okay, thanks so much.
Mm-hmm.
And at this time, I'm showing no further questions.
Thank you.
Okay.
This is Irene right here. Tim, did you?
We have a little bit of time, so we can just wait a minute or two, and if there are no further questions, we can give it back to you, Irene. If that's okay. Maybe a minute, not too long. But we're available and ready to answer questions.
And as a reminder, if you would like to ask a question at this time, please press star 1. We have a question from Kodamoti Venkat. Your line is open.
Yeah, thanks for taking the question. My question is for serology tests, just like how NCI is doing, has been doing, looking for the binding antibodies, or NCI or any other institution will also do the vaccine antibodies. Do you have any plan for having a center that can do independent validation for antibodies that can show either neutralizing antibodies or protective antibodies?
We recently issued a statement on use of serology for vaccinated folks and people who had a natural infection, and the implications currently for what can be said about protection or immunity, which the studies, to our knowledge, haven't been done to support that use. Potentially both neutralizing and non-neutralizing assays could be used. Most likely, truly quantitative assays are going to be the assays that are going to provide the most important information about that. If things go as we hope, that it's not just the presence or absence of antibody for other examples where the tests have been authorized to assess the vaccination, but it's the level of antibody sometimes that is important. It's an obvious area for us to look into getting additional information about the serology banked samples at SCI, having to do with quantification of antibodies and the presence of or absence of neutralizing antibodies. So it's certainly an area of active discussion right now. But in all, because the focus may be more on truly quantitative assays, those are usually not point of care assays, although I'm not going to rule it out. So it may be something that we eventually design a blinded panel for, if we can, depending on how soon we can stand up such a program. But I can't make any promises that one will be stood up, and if it is stood up, how long it will take to stand it up. But it's an obvious area for consideration right now.
Thank you. But I think in the past, when you were mentioning vaccination is not diagnostic, so do they really need the approval, the way in which it is done, and even I think it was mentioned that it is not CDRH, it is CBER, and we will discuss. And also, can you just mention still it is a part of a CBER thing with vaccine and vaccine efficacy, or it is not a diagnostic test?
Oh, well, it's very clearly a diagnostic test. It's just that it moves into CBER sphere as well. And we have agreement with CBER that we will combine our efforts to review applications that have to do with, particularly that have to do with any claims about immunity or protection. That's the situation we're really talking about here. All other types of assays that don't speak to immunity, or all other serology assays for COVID that don't speak to immunity or protection would be reviewed within our office and our center.
Thank you.
Mm-hmm.
Our next question is from Lonnie Edelman. Your line is open.
Hi, thanks. Good morning. I actually haven't called in a few weeks. It's good to hear all the questions, and thanks for continuing the discussion. There was no template for a reader to get an EUA, so we submitted a pre-EUA a couple months ago, maybe 3 months ago. I was wondering how the flow is going. You had a surge going at the beginning of the year. Are you? I guess what I want to know is, are you going to get to the pre-EUA anytime soon, or are you still really backed up?
So I think, sorry for the delay in getting back to you. And certainly I'm not sure that I'm aware or Toby's aware of your pre-EUA. So if you could forward that to our template email address and make sure that your submission a few months ago is seen by Toby and I. We will look into it and we'll get back to you. One of us or one of the team members will get back to you. One thing with readers is, in order to assess readers, we do recommend that it comes in with a candidate test.
Sure.
And that's how you assess it. So that's just for general. I know what you put in your submission, but that's just for a general consumption. That the only way we can evaluate a reader is in the specific application in use with a specific test.
Yeah.
We just don't know on the readers how portable they are from test to test, right? And we clearly have seen some issues with some readers, and we're not quite sure why. It could be, individual software applications aren't robust enough. But for example, we've seen variable performance across different makes and models. That gives us pause. And if something isn't robust enough to work across several makes and models, it may not be as useful because there's a lot of makes and models of smartphones out there if you have a smartphone application. And we do want to see a validation plan that incorporates diversity of smartphone and even software version use on those smartphones for obvious reasons.
Yes, I'm in full agreement. The system was designed with a captive phone. So anyway, I don't want to turn this into an infomercial. So we'll send in the email and hopefully. Yeah, I will.
And Toby and I will make sure you get a response.
Great. Thanks again for continuing to do these meetings. It kind of feels like things are straightening up, and maybe there's no need, but there's still a need, at least for us.
For developers, I completely agree, and I have recommended to FDA leadership that for non-COVID, after we're done with COVID, that we have some kind of forum like this for all IVD developers. It probably won't be a weekly call. It probably will be a monthly call. But
Great.
My desire and the FDA's desire is to give ready access to developers to ask questions and to make that as easy as possible. So it's part of our overall customer service
Yeah, that's great. Yeah, good to hear. Thanks so much.
Mm-hmm. You're welcome.
Our next question is from Ashwood. Your line is open.
Thank you. I'd just like to build on the statement that you made last week on antibody testing and on the fact that all currently approved ones should not be used for verifying immunity from vaccines. Do you foresee a series of new tests, or are they already in the pipeline that would be vaccine specific, i.e. they would be detecting specific antibodies to the Pfizer or Moderna or Johnson vaccine, and would they need to be labeled to be matched with their vaccines?
Yes, I don't have a crystal ball that tells me how this is all going to work out. It's my hope that measuring antibody response to vaccines and post-infection, natural infection, will afford some ability to speak to immunity and protection based on clinical study data. Clinical studies are being performed. They're obviously easier to perform relative to specific vaccines because you know prior history because of in those studies, because you've done the investigators have done serology testing to know prior to vaccination that they haven't been exposed and aren't currently positive through a molecular test when they get vaccinated, and then you know exactly when they get vaccinated, so you know the start of the immune process to raise antibodies, the level of antibodies, whether or not there's neutralizing antibodies present. So these studies, you know, in determining outcomes from vaccination or any sort of obviously infection are the easiest to carry out in the vaccinated population for obvious reasons. And I hope that we don't link it to a specific vaccine, but that we link it to something that is can be universalized. This is my hope. There's no promises, but say a certain level of binding antibody to to the appropriate antigen. With the use of the appropriate antigen and a serology test that cross-reacts with vaccines, and most all the vaccines all the ones authorized in the U.S. are spike protein vaccines right now. I hope that there's standardization through the international WHO standard, so that we can say, this cut point or this level of antibody and above is protective. And we can roll that out as for use by developers in updating appropriate tests without them having to do their own outcome study with their own tests. So the only way I can think about doing it now, and I'm open to other ways, is to actually utilize the WHO standard to link the study assays used in these clinical trials to the international standard, and then from there go back through the international standard for any other developed tests. I think it would be ideal if we had a bank of immune plasma serum that could also be used to perhaps send out and blindly test developers as part of the process, or perhaps as part of the post-market authorization process to doubly ensure that the information that's being assessed is accurate and useful and can be counted on. So it's a long-winded answer, complex area right now, but one of obviously intense thought within the FDA.
All right, thank you.
And at this time, I'm showing no further questions.
And we're at the end of the hour, so it's a good time to turn it back over to Irene.
Thank you. This is Irene here. We appreciate your participation and thoughtful questions during today's town hall. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training www.fda.gov/CDRH Learn by Wednesday, June 2nd. If you have additional questions about today's presentation, please email cdrh-EUA-templates@fda.hhs.gov. As we continue to hold these virtual town halls, we would appreciate your feedback. Following the conclusion of today's virtual town hall, please complete a short 13-question survey about your FDA CDRH virtual town hall experience. The survey can be found now on www.fda.gov/CDRH webinar. Again, thank you for participating, and this concludes today's virtual town hall.
Thank you for participating in today's conference. All lines may disconnect at this time.